This video is about lymphocytes, and lymphocytes are the adaptive immune system. They are what make it up, and you're going to learn a lot about lymphocytes and specific lymphocytes. This slide shows up 3 different times in this set of slides. This is the first time you'll see it. I put it in 3 different times because I don't want you to miss it. It is so important. You will not be able to understand the adaptive immune system if you don't go through this slide and understand this slide. So, let's get into it. Each lymphocyte, so this is 3 different lymphocytes, each one is covered with 10 to the 5th, that's 100,000 identical copies of one unique T-cell receptor, or if it's a B-cell, B-cell receptor. And each receptor detects one epitope. So let's just stop there and think about that. These cells are drawn to illustrate this, but instead of 100,000 copies, they have 3. So this cell, this lymphocyte, has 3 copies of one receptor. Each copy is the same. These are B cells. This is what a B cell receptor is sort of designed like. It's a Y-shaped thing with 2 receptor parts that can bind something. And so this one is drawn with little squares. The idea is that the epitope this cell can detect is a little square thing. Realistically, it would be some very complex part of a protein, but it's best to learn it with shapes and simple geometry. Okay, so this one has 3 copies of the receptor that detects squares. This lymphocyte has 3 copies of a different receptor. It detects little circles. This lymphocyte has 3 identical copies of a different receptor. It detects little triangles. And so, whenever you're studying adaptive immunity, start here. Start by drawing lymphocytes and just reminding yourself, okay, a lymphocyte is mostly nucleus, honestly. That's what a lymphocyte is. It's a cell that is full of a nucleus, and it's also going to have several copies of one receptor. So let me be nice and fill in the nuclei. It's so boring to draw nuclei in a lymphocyte. Okay. In that slide, they were B cells, but these will be T cells. And T cell receptors are drawn like this. T cell receptors have a different structure from B cell receptors. T cell receptors only have one detecting part. And the way I'm trying to draw it is this one detects circles, this one detects little triangles, and this one is going to detect little squiggles. It would be nice if I were better at drawing, and it would be nice if the software made sense to me, but it doesn't. So what are you going to do? So this is really a worse drawing than I thought I would make, but what I am reminding myself by drawing this is that each lymphocyte has its own one receptor it can make, and it makes many copies of it. This lymphocyte makes many copies of its receptor. It has one different receptor it can make, and it makes many copies of it. So really the way to think about this is that in the DNA, if I'm just drawing DNA of every lymphocyte, of every cell in the body, really, you have the same DNA as every other cell. Every cell has the same DNA as every other cell. There are these little mini genes, and I'm just drawing little squares because what this means is that each of these little squares represents a short segment of DNA. And so we could say this is region 1, and then region 2 there are lots of other little mini genes, just short pieces of DNA that are kind of separated from each other. In region 3, there are lots of little short mini genes. And what happens is when a lymphocyte is young and very immature, right when it's being made in the bone marrow, what it does is it chooses randomly one of each. And then it kind of scrambles them. And what comes out of that is the gene for a receptor. So this is a way that our lymphocytes can randomly generate a new receptor. So each lymphocyte gets its very own randomly generated receptor. No one knows what it detects. So I drew this detecting a circle, but that's simplistic. What it's going to detect is some part of a protein. That protein might not even exist, but it's ready to detect it if it does exist. And this is going to happen when a lymphocyte is very young, and it's going to pass down the gene for making that receptor to its daughter cells. And each daughter cell is going to have the exact same receptor. And their daughter cells will have the exact same receptor. So you'll have a lineage of T cells that have this exact same receptor. And this T cell will have a lineage of a different receptor. So that's really the essential thing about lymphocytes, is they have many copies of one randomly generated receptor. So each lymphocyte can only see one epitope. This lymphocyte can only detect one epitope. It has 100,000 copies of that one receptor that can only detect that one epitope. This one can only detect one epitope, and it's a different epitope. Nobody knows what it is. It might not exist, or it might be part of a SARS-CoV-2 spike protein. It might be one of the most important epitopes right now. Who knows? And this one randomly generated its own. That's the only thing it can see. So lymphocytes, am I right? They're able to make, through shuffling the DNA, an astronomically huge number of different possible receptors. Now, we don't have this many lymphocytes, so we don't have all of these. But when they are randomly generating the receptor, they get one of, this is 10 trillion. They get one out of the 10 trillion possible receptors. What this means, realistically, is that any protein that will ever enter your body, there is probably a T cell and a B cell that can detect it. And that's amazing. So even if it's a protein no one has ever imagined before, no, it's never been made on Earth before, if it's made, chances are there's already a lymphocyte that can detect it. And if so, that's the only thing that lymphocyte can detect. So that's lymphocytes. So they do this recombination thing. That's what I just walked you through. And so they have a unique way of distinguishing self from non-self, and it is that they can't. All they can see is whatever their receptor sees. That's the only epitope they can respond to. And something we haven't gotten to yet is like, why don't they detect our own cells? Why don't you have a lymphocyte that has a receptor for actin or myoglobin or myelin? You don't, because those lymphocytes, they were generated. They did exist, but during lymphocyte education, they are killed so that your lymphoid tissues are full of lymphocytes that cannot detect any protein made by a normal human. So ideally, no lymphocyte can detect a self epitope. So they are blind to self antigens and self epitopes. So a cell is an adaptive cell if they all have these characteristics. If it depends on a randomly shuffled DNA to make a protein, and that's the protein that it depends on to do its job. So that's how antibodies are made. The genes encoding antibodies are randomly shuffled to make a new antibody when a B cell is very young. T cell receptors are arguably more important. T cells are really the critical thing. As an aside, antibodies prevent you from getting the virus. T cells defeat the virus if you get the virus. So that's really how to think about that. T cells are very important. So the T cell receptors are the result of randomly shuffled DNA, same way. So a cell is adaptive if it has those randomly generated receptors, and only one, many copies of one identical randomly generated receptor. And it's adaptive if it does not use pattern recognition receptors. So a lymphocyte doesn't use pattern recognition receptors. It uses its one very special receptor. So a cell is adaptive if it is a lymphocyte, in other words. For our purposes. Okay, so the next video will be about antibodies. Antibodies are important. They can prevent you from getting a virus. They can do a lot of other things that will save your life. So that's cool, and we will learn about that in the next video.